Let me get started then. I hope a few more people will join us today. Thank you for coming back after Thanksgiving to our virtual seminar series. I know a lot of people watch this on the streamed version, so I hope there'll be some good questions for our speaker today. Today is the last day of November. It's getting to the end of the year for the working group. And I want to remind everyone, as always, that this meeting is live streamed and recorded and made archivally available after the fact. As always, Tom Helikar and I are available for comments and suggestions, as well as Jim Sluka and Lorenzo Vischini. Some of the administrators are not here today for one reason or another. Tom is in Barcelona teaching, and Lorenzo is also traveling today. And they send their apologies for not being here at the meeting. We have our communication channels. It helps if people help us keep these up to date. We have the LinkedIn page, the IMAG Wiki, our YouTube archive of videos, the X MSM viral tag. Please help us let people know about the resources that we have made available for this community. We have the GLIMPRINT handle now. Please do help us build that. We appreciate any help that you can give us in creating a community that is vital and successful. Again, the MSM viral tag on X. I still have trouble not calling it Twitter as well, formerly known as Twitter. Please do let us know and help us also advertise the talks once they're archived. Some early announcements. There is a staff scientist position in neural developmental dynamics at NIH that may be of interest to this community. There are a number of funding opportunities in Europe. People have seen some of these announcements before. CDAI, I think is the pronunciation, single electronic data interchange area, accelerating implementation of new approach methodologies, which coordinates very well with the new NIH initiative in that area, and I know many people in the community are interested in. Maximizing potential synthetic data generation. January 2024 is getting pretty soon for these large-scale EU proposals, but maybe there'll be additional announcements coming up. Another next-generation proposal with a slightly longer deadline on scientific instrumentation, but that also includes software development. Innovative health initiative as well, also early deadline. We will have our regular steering committee meeting tomorrow morning at 11 a.m. If you think you should be invited and you don't have an invitation, let me know and we'll send it to you. The topic of discussion will be primarily organizing our first annual meeting for the community. Are there any other announcements that people would like to make before we move on? Okay. Coming up on the 14th of December, we have Tongli Zhang from University of Cincinnati talking about medical digital twins. We won't meet on the 28th. I don't think anyone wants to meet between Christmas and New Year's. We'll get started again on January 11th. We have Jake Beal, who's done some really interesting work on crowdsourcing, very heavily involved in the iGEM program. We have Adam Hollis, who's done modeling of angiogenesis and cancer. And then we'll have, going forward, AI-based computational modeling to support drug development on autoimmune diseases. If you have suggestions for speakers, please let us know. If you've spoken before, you can always speak again. Suggest your students, your postdocs, people that you would like to hear, so that we can keep the series going and make it of interest to our community. With that, I don't want to take any more time for our speaker. We very much appreciate his willingness to step in on short notice to tell us about what's going on at Certara, and I will turn over the share to you.
Well, thanks for the introduction. And yeah, excited to speak to you about my talk, which is titled Predicting Disease Activity Scores and Inflammatory Bowel Disease with Simulated Tissue Biomarkers using QSP. I'd just start by acknowledging my colleagues at Certara who helped develop this approach, including the model, as well as our collaborators at Sanofi. So what I'm hoping everyone gets out of today is a conceptual understanding of the type of modeling we do, which is quantitative systems pharmacology, which you may already be familiar with, and also the key application of QSP modeling that we are proposing. In today's example, we'll be using inflammatory bowel disease, but we do believe this can be used generally across different disease areas as well. So a brief history. So QSP modeling is really a bridge of two different styles or disciplines. First is the pharmacology modeling, such as pharmacokinetics, pharmacodynamics, as well as physiologically based pharmacokinetics, where the discipline really looks at the target exposure, the engagement, as well as the modulation of the target. And we combine that with the systems biology modeling that we do normally for different disease areas, which includes the pathways specific to the target. That could be intracellular as well as organ level pathways. We've also tried to model the pathophysiological regulation of the disease and also the modification following the intervention by the pharmaceutic. So here at Certara, we developed a platform approach. We developed a platform approach where we combine different modalities such as bispecific or multispecific antibodies, gene therapies, immunogenicity such as anti-drug antibodies and autoantibodies, vaccine therapies, as well as lipid nanoparticles. So it's a quite flexible approach that can accommodate various different disease or therapeutic modalities. We combine that with the disease platform, such as neurodegeneration. We've modeled Alzheimer's, Parkinson's, ALS, immuno-oncology, as well as hemophilia. And lastly, the pathway or system platform. In today's example, we'll be focusing mainly on the cytokine and immune response in the gut. So things like TNF-alpha, IL-23, IL-6, and interferon gamma. But we can also focus on other pathways such as a complement system, neuroinflammation, IL-2, and also other IO targets such as checkpoint inhibitors. So altogether, we've created an integrated QSP platform, and the goal is to be able to simulate in silico trials and experiments to help guide drug development. So what we believe the killer application for QSP moving forward is based on the clinical endpoints. So as you may already know, QSP is predominantly used for clinical trial design, such as dose finding studies, recommended phase 2 dosing. And generally, the way that's done is by connecting the dose of the drug with the biomarkers for a specific disease. But what we believe is missing as the next big leap potential for QSP is to begin to predict actual clinical endpoints. So I'll go over the example today I'll go over extensively is on inflammatory bowel disease. But things like how the patient is feeling, overall survival rate, these are more difficult to achieve in a mechanistic level. As you may know, there are other more model-based meta-analysis that can be done to help predict those studies. But what we believe is that if we, depending on the disease area, we can connect the simulated tissue markers to the actual clinical endpoints using this approach. So again, today's example, I'll go over inflammatory bowel disease and how it connects to the biomarkers. And then we'll use elements of artificial intelligence and machine learning to try and connect the response of those biomarkers to the actual clinical endpoints. So we've built an IBD model, QSP model, and it's based on the what we did is we took a comprehensive approach to try and predict the disease activity scores. So initially it was based on the Pfizer model, which is from the Rogers 2021 paper. It has a mechanistic multi-state mathematical model that includes the key biological mechanisms in the blood and gut, cell differentiation, cytokine production, and various clinical biomarkers. The diagram below shows you the extent of the various different immune response that we've modeled. And the key thing here is that the model primarily focuses on inflammatory mediators, the key immune response that occurs during Crohn's disease and ulcerative colitis. And the nice thing about this publication was that I was able to represent both Crohn's disease and colitis using the same model with the same underlying pathophysiology. You'll notice that we simplified or focused certain elements of the modeling. For instance, we don't have an extensive representation of the epithelial cell lining inside the intestinal lumen. We don't have extensive representation of the epithelial barrier function, which also has been implicated in the disease. But primarily, we're focusing on reducing the inflammatory response once the mucosa or the lamina propria of the gut is exposed to bacteria, other types of physical injuries that occur during the course of this disease. What's important is also that we've represented a blood compartment so that we can correlate our model and start to validate using serum level measurements such as CRP to get a sense of the overall level of inflammation. We have both innate immune response as well as adaptive immune response, primarily T cells that are recruited from both the local lymph nodes, such as the gastric and mesenteric lymph nodes, as well as more systemic lymph nodes and recruitment of T cells or T lymphocytes. But primarily the disease, as we know, is driven by the myeloid cells, so things like macrophages, particularly M1 phenotype, as well as the neutrophil activity. And that's predominantly why the key biomarkers that's measured throughout the course of this disease are fecal calprotectin, which is released by the neutrophils, as well as serum CRP, which gives us an overall sense for a level of inflammation. Another thing is we've included an extensive list of cytokines, chemokines, and other pro-inflammatory mediators in the hopes that in the future this model could be used for other additional target explorations. The two particular therapies I'll be showing you today are adalimumab, which is an anti-TNF monoclonal antibody, and mirikizumab, which is an anti-IL-23 antibody. So with this model, we were able to investigate the right target and find out the right dose and also the right action, which is predominantly based on the baseline measurements of a patient. But the most exciting part is that we're able to predict the clinical scores. So with this current implementation, we're able to predict the Mayo Score for colitis patients and the Crohn's Disease Activity Index or CDAI score for Crohn's disease patients. I'll go into more detail on how that's done in the following slides. So this just to show that we're able to predict the clinical scores and the clinical response quite well using our virtual populations. So examples here shown are the adalimumab and mirikizumab at 2 different dosing regimens at 4 different clinical trials. So the nice thing about IBD is that there are plenty of published clinical trial data that we can use to both verify and validate our approach. So we look at week 12 and week 56 measures of fecal calprotectin and serum CRP at various time points to make sure that our virtual population that we have developed captures the biology quite well. So we take also a look at the baseline measurements of these patients and the treatment response overall with time. And what we found is that the model is able to create virtual populations that match the expected distributions for several published clinical trials, including clinical remission, which may be defined differently from trial to trial, but in examples I show you today will predominantly mean a CDAI score of less than 150. So this is giving you a bit more in-depth idea of how the model simulates the various cytokines and immune cells populations. So it's important to see that we're able to have a diverse amount or diverse baseline levels of these various cytokines and cells, as well as diverse treatment response following the various therapies. And further, we can start to separate out patients who are responding to particular therapies versus those who are not responding. So in this example, on the top, we showed an ulcerative colitis virtual population, where on the left, we have responsive patients. You can see predominantly that the level of the inflammatory cytokines are significantly inhibited. And on the right side, we have those patients who we classified as non-responders to this particular therapy. And we see that the treatment is not able to effectively inhibit some of these inflammatory mediators. So we do have data from actual patient biopsies on what the levels of these mediators should be at baseline. And we have understanding of the various interactions of the immune cells and the cytokine regulation from in vitro studies, as well as some animal in vivo studies. But predominantly, we're not able to measure what the cytokine immune cell levels should be during treatment or even after treatment for many of these patients. So it's quite an interesting inverse problem we have, where we understand their clinical response according to these scores, as well as some key biomarkers like calprotectin and CRP. But largely, we don't know what's happening inside the gut mucosa for many of these individual cytokines. But this is where we think there's tremendous opportunity, because if we can simulate with these virtual markers, we'll then be able to try and tease out a relationship between what's happening inside the mucosa and how it translates to actual disease activity and clinical scores. So this is just a brief overview of the type of clinical scores that we are trying to predict using our model. I started off with the score that we consider the most mechanistic out of all of them. And what I mean by that is that the Mayo endoscopic score, which is performed by a physician during an endoscopy, these scores tend to correlate quite well with actual inflammatory markers. So we wanted to start off with something that had a strong connection and then branch out from there into some of the less precise and more qualitative disease scores. So this is just an example. On the left side, you see a Mayo score of 0 essentially means that the mucosa is healthy. There's no sign of disease. And as the disease worsens, as you go towards the bottom, we see some erosions. The epithelial lining starts to become compromised. There is less vascularization, and ulcers and bleeding starts to occur. So this is primarily what the physician is looking at and to assess the disease. But what we want to do was to predict this using the simulated markers of the cytokines and immune cells inside the tissue itself. So I'll give you some example references that we had used on our right side. There have been a number of studies trying to connect the level of T cell activity, maybe some cytokine concentration levels from biopsy samples with the severity of the disease. The reason why this is challenging, though, is that a lot of these papers are not coordinated. And what I mean by that is that these patients are different. It'd be nice if for a single patient we measured their full spectrum of cytokine profiles and cells, but instead what we have is these separated studies that try and establish some level of correlation between the disease severity or disease scores with things like Treg levels, IL-17 levels, TNF alpha, and so on. Well, luckily, again, for us, there is a large volume of literature trying to establish these relationships. But the challenge here, because we don't have individual level measurements and because these are coming from various different patients, is that we cannot mechanistically connect these markers with those disease scores. So that's why we decided to employ some elements of AI and ML to try and understand and figure out some of those relationships. So what this table summarizes is the volume of literature that we had found establishing the roles of the various immune cells and cytokines and how they relate to each of the disease activity scores. We've looked at a long list on the left side, but ultimately, as I mentioned, we started off with the Mayo endoscopic score because that seemed to have the most mechanistic link between tissue level inflammation and the actual disease score that's assigned by the physician. And then from there, what we're able to do is use all of this as training data for an AI approach. So if we can understand the relationship, it's a combination. So I'll go into more detail in a bit later, but there is an element where we have to use scientific expertise to understand what the key features are that we should focus on. If we allow the AI training to decide which features are most important, chances are it'll end up with some unclear relationship. But what we want to do was base it on actual physiology from scientific expertise, knowing that things like interferon gamma has effects on vascularization, and therefore that would have a direct effect on the score that's assigned by the physician. So those types of known mechanistic links we leverage from in-house expertise. And then from there, we use some of this data to try and estimate the parameters of the model that would be able to assign or categorize these patients according to these scores. I think something that's interesting here I'd just like to point out is there's been a lot of activity around Treg research regarding IBD. But if you look along the column, we'll see some conflicting evidence where sometimes it states that Tregs are actually correlating with severe disease, whereas other research says Tregs are inversely correlated with the severity of disease. So I think there needs to be a little bit more work on whether that is locally induced Tregs versus natural Tregs versus more diapedesis of Tregs that have been activated from other sites and other organs. But I think that was notable. And again, this kind of gives a reason for why we need to have more of a AI machine learning approach to understand these relationships. So this is just a conceptual formula. It's not the final formula that we had used to predict the disease scores. I want to give you a sense for how the scientists thought of the problem. So for example, the Mayo endoscopic score, they try and use something like a Emax function or a Hill function to relate each of the various immune cells and cytokines. But what we had found was that we're essentially dealing with categorical data. Whereas these functions are trying to create a smooth function to assign a score between 0 to 4, what we needed more was a classifier that classified a patient based off of various markers. And this gives you, again, a sense for what the scientists believed were predictive of the severity of the disease. So things like interferon gamma, neutrophil activity, Treg activity, and epithelial damage. And then from there, we can use the endoscopic score to add on to other elements to try and predict the total Mayo Score, which is a more commonly used clinical endpoint in these trials. And again, we expanded it, or the scientists want to expand it further by introducing TNF alpha, IL-8, and T helper cell type 2 as part of the prediction. And there was some data to support these indications. So on the left side here, you see there is maybe a loose positive correlation between TNF alpha levels and severity of disease. It is classified with a CDAI score. We see some correlation between CXCL8 or IL-8 mRNA levels with clinical index scores, and also IL-13 positive, FOXP3 positive, which, this kind of shows you the plasticity of the CD4 T cells that we're dealing with. But we used what we consider the standard markers based on, again, the in-house scientific expertise on how we classify these T cells and then their relationship with each of these scores. So if you look at any one single source of data, it's hard to say that there is a strong correlation or let alone a causation. But we look at the full body of literature evidence and use that as a training set, and we start to see a bit more precise picture starts to emerge. So here's an example of a strategy we use for the Mayo Score prediction. We started off by looking at preparing training and testing data sets for building this prediction model. And the way we did that was, again, based mainly on the literature, as well as some in-house data, where we're able to establish a relationship between the total Mayo Score and Mayo endoscopic score with the key biomarkers such as calprotectin or serum CRP. From there, we sought out to build a regression model based on two different strategies. So in the first strategy, which is the most straightforward, we want to establish a linear relationship between the tissue measures and the Mayo Score. So in this graphic on the bottom left, we would extract out the TNF alpha, interferon gamma, and other cytokine and immune cell levels from the virtual patients in our platform, and then use that as training data to try and establish a relationship with the clinical scores based on the biomarkers. So the biomarkers were used as a mapping, but ultimately what we're trying to establish is individual cytokines and immune cells and disease activity scores. We want to eliminate any unnecessary predictors, so we use regularization, so lasso technique, to try and zero out some unnecessary features. So we started off with a large feature set that the scientists believed were implicated in disease activity and severity. And then from there, we pruned down the ultimate, the final list of features. And another concurrent step, we use more of a categorical regression, specifically a multinomial logistic regression model for calculating the Mayo endoscopic score. And again, we use lasso regression to generate the total Mayo score. So in preparing this training and testing data, we have to build a limited individual patient tissue measurements were taken from the literature. So again, the biomarkers and the disease scores, and we connected that with the individual cytokines and immune cells in these individual patients to map ultimately to the Mayo score. So the strategy here was to use fecal calprotectin and CRP and its relationship with the Mayo score to try and impute what the patient gut measurements would be by using the Rogers model virtual population. So use both fecal calprotectin and serum CRP to filter out the Rogers virtual population, ultimately linking the selected virtual patient with the relevant Mayo score, and then we would impute the gut measurements after that. So here's just a glimpse of one of the prototypes we had built to try and assign these Mayo scores. So if we look at the Rogers VP that was provided in the paper, we classified those patients with a score of 0 to 3 based off of the multinomial logistic regression model. And we found that we're able to have a prediction accuracy of around 60% or better for most of these scores. Right, so what we have found was that typically in these clinical studies, they measure not just individual scores, but the level of remission or regression is calculated by having a score of 1 or less. So if we actually combine some of these categories together, we can have even greater accuracy in our predictions. This is just one iteration of our approach. It showed that it is better than what we had previously have done, which is based off of the different inflammation, we look at the biomarkers, and we were having around predictions of 40%. So already the AI approach or the multinomial logistic regression model was learning something about the features. So now this is a diagram showing you how it actually looks for our virtual patients. As mentioned before, we mapped the simulated gut markers from the QSP model to the clinical biomarkers, and then over to the disease activity scores based on various literature data. And then on the top, on the right side, we start to see how this looks like for virtual patients. So for a single virtual patient shown here, the black line is the untreated simulation, and the color lines show the treatment response to an anti-TNF and to an anti-IL-23. So based on the virtual patient and based on the type of treatment, we're able to predict now the Mayo endoscopic score with various probabilities using the multinomial logistic regression approach. So in the untreated scenario, we predict that this particular patient would have a 100% chance of being classified as 3 or severe disease. And then following various treatments, the anti-TNF, for example, there's a 75% chance that patient reduces down to 1, which would be considered clinical responder, or with mirikizumab, a slightly worse response, but still an improvement of a score of 2. But with the combination therapy, there's a good chance that patient will be classified as 0 or a complete clinical remission. So this starts to get interesting because although the adalimumab and mirikizumab, these are published trials, and we can match this exactly to data to make sure our model is behaving appropriately. What's not been done before is a combination of therapy of adalimumab and mirikizumab. So we can start to test out untested scenarios, untested dosing regimens to see what the potential of various therapies could be. This is just another example. So in a previous slide, I showed you the response of an individual virtual patient. In this example, I'm showing you the Crohn's disease virtual population. So we've created a population of about 100 virtual patients that matches several clinical trials. And what I'm showing you here is the overall readout of this Crohn's disease virtual population and the key biomarkers and the disease score. So based off the literature, you'll see that we have close agreement with the baseline and the week 12 treatment response for these different measures. And we're also able to predict about a 66% response rate, which is quite close to the 62% response rate. So there's a fair amount of verification, validation work that went into this to make sure that our predictor was behaving appropriately. And on the left side, you see here, this is showing you the full virtual population. You can see their baseline measures, week 12 and week 52, and how these key biomarkers behave, as well as the distributions. And then for the CDAI score, and again, we use a categorical model. So a green would mean a score of less than 150 or remission. Anything above 150 up to 220 is classified as teal. And then anything worse than that would be blue, which is severe disease. So at baseline, we see that all the patients are classified as either moderate to severe. By week 12, 66% of the patients are become clinical remission following this therapy. And at week 52, we see a slight drop off during the maintenance phase, where some of the responding patients worsen back to moderate disease. So as I mentioned, we can start to test some untested scenarios and dosing regimens. So we start off by validating our virtual population treatment response rates. So for example, adalimumab, 40 milligrams Q2W. Based on this reference, we see about 50% clinical response. We ask the question, what if we doubled the dose size? And what would happen? So we see that we predict we have an improvement in the amount of patients who would be classified as clinical responders. We did the same exercise with 2 different dose ranges for mirikizumab, and again saw an improvement in the percentage of patients who respond. And then we tried something unique where we simulated the combination of the therapies with different dosing schedules, and again saw a moderate increase in the amount of responders. And on the bottom here, I'm showing you the various simulated markers in the model. And again, you can see that we have diverse baseline measurements, and following therapy, there is differences in the treatment response as well across these different markers. Okay, so here's a little bit of a unique approach. So most of the what I've shown you was based off of dose finding studies for late stage clinical trials. We had a recent collaboration. We want to understand how do these patients respond to a very early stage compound. So this is in the early target validation, candidate selection, lead optimization stage. And they're interested in a kinase inhibitor for ulcerative colitis and Crohn's disease. As we know, these inhibitors these kinases mediate tissue damage. So there's a particular target that our collaborator was going for, and based off of the profile of the compound as well as the pathways, we implemented an inhibition of these various pathways in our model. So TNF receptor 1, IL-1, toll receptor, T cell receptor, and TGF-beta for up to a year. Now, this is very early stage, so we did not have any PK data or PD data. We were going off of a fixed inhibition level of these various pathways in our model to see how our virtual populations that we've already validated with other therapies, how they would respond. So we saw that the various markers like TNF, IL-6, the dendritic cells, and T helper 1 cells do respond quite well based off of the percent inhibition in the model. And interestingly, what we simulated was that with an approximate 50% fixed inhibition in the in vivo potency of their drug, we're able to see a decrease in IL-6, macrophage 1, and CD4 levels at a significant rate. And ultimately, that at 16 weeks, we predicted that there'd be about a 61% remission rate, or a CDAI score of less than 150, if this drug is able to reach the appropriate exposure and also inhibit the target by up to 50%. So we don't know yet if that would be the case, but they want to have a very early indication of whether this would be a promising approach. And it turned out it was. But alternatively, what they also want to understand was, would their drug work for adalimumab inadequate responders? So as you may know, there's a significant fraction of patients who do not respond to anti-TNF cytokine therapy or anti-TNF antibody therapy. So you want to know if maybe this could be a follow-on for those patients who are unresponsive. But we had found was that only about 3% of adalimumab non-responders would actually respond to this type of therapy. So not much promise there, but overall the remission rate looks good. And also we want to understand a recently published study. So as you know, JAK inhibitors have now been, studies have been conducted for Crohn's disease patients. So we want to know what level of inhibition would a JAK inhibitor need to achieve in order to match the clinical remission scores that we saw in this particular study. So based off of the type of JAK inhibition, which was a JAK1 inhibitor, and based off its selectivity and potency, we simulated an inhibition of these following pathways in our model. And then we saw the breakdown of our virtual populations between responders and non-responders to this simulated JAK inhibitor. And what we calculated was that based off the study, they only achieved about 20% remission, which doesn't seem very promising. When we look at our simulated markers, again, we only see about 10 to 15% drop in inflammation. And overall, with our model, we found that the JAK inhibitor seems only to be inhibiting these pathways around 15% on average. Again, this is just an estimate because we're using a fixed level of inhibition and not the actual PK and PD of the JAK inhibitor, because that's yet to be published. But I think this gave the client a good idea of what can be done, because if they can achieve 50% inhibition, they'd have a strong result. But with the JAK inhibitor, it seems they only achieved about a 15% inhibition in vivo. So it's just something to compare against for their future program. Okay. So as I mentioned, we believe this approach can be extended to other disease areas and other existing QSP models. I've mentioned to you about inflammatory bowel disease in today's example, but we think that with neurodegenerative disease and other indications, what we can do is start to connect the QSP model with the clinical data. So we have a database of published clinical trials, as well as some proprietary trials in our database, which we can connect with our QSP models. And then from there, we can use more general AI approaches. Currently, we're developing GPT, large language models, to try and annotate and build these biological maps in an automated fashion. But this could be, in our opinion, repurposed so that we can start to predict more and more of these clinical endpoints using the simulated markers within QSP models. Thanks for joining and thanks for your attention, and I'm happy to take any questions.
Okay, thank you very much. Questions from the audience? John, go ahead.
I look at the effort to develop modeling tools that can be used in clinical trial applications, and I'm the naive member of the community here, so the question may not make sense. But if I understand right, this model was able to predict the clinical outcome of a sample of people who participated in an actual clinical trial as the source of validity.
Mhm.
In the clinical trial world, there's a whole lot of other questions that also need to be answered, like, are there side effects? Are there adverse effects? Is there anything in the way this type of model worked that hinted or suggested that in some cases there would be side effects to some degree of severity? Okay, you looked at the clinical diagnosis after treatment and say the model predicted that in numbers.
Mhm.
Okay, I don't understand whether it predicted it by individuals that made up the numbers or just the numbers themselves, percentages.
Sure.
And is there anything in the model that suggested that some of the participants in the clinical trials had various kinds of adverse effects?
Sure. No, yeah, that's a fair question. And there are, some serious adverse events that do occur. So patient dropout is a real thing in IBD. So to answer your question, the model does contain the elements to be able to represent safety issues. So things like cytokine release syndrome, let's say, or neutropenia. We have these cell populations and cytokines represented, although I hesitate to say the model can predict that because it hasn't yet been validated to safety data. So its primary use was focused on just efficacy for various compounds. Now, there are other models out there within IBD as well as many other disease areas that focus on toxicology or safety issues. So that's quite interesting work as well. And your other question is actually a very good question. So depending on the sponsors that we work with, we may or may not have access to individual level data of patients. Now that's very key because we need to know the full cytokine picture of an individual patient, what their score was, and how they respond to treatment. Now, the part I'm showing you today is based solely on publicly available literature, which means it'll only be distributions of patients. So without that individual level of knowledge or information to tune each patient, like digital twin approach, let's say, without that, what we can rely on, though, is to make sure that a single collection of virtual patients, let's say our 100 virtual patients, are first off, physiologically, they make sense, right? There's nothing, they're within range of actual published human biopsy samples from inflamed regions of their colon or their ileum, let's say. So that's at the baseline. And as for treatment response, if we can match several clinical trials simultaneously, as I showed in that one slide, we had 4 trials that were able to match using the same 100 virtual patients. That starts to give us a bit more confidence that our parameterizations are within reason, definitely within the distribution, fits within the distribution of several trials. But again, doesn't quite match yet. The gold standard would be individual level data.
So then one other quick follow-up. Were there things that the process of developing a model or the application of the model and the validation that were discovered about what was going on between the pharmacology and the biology, that the kind of things that we don't know, we can't discover in clinical trials very well? Were there any examples of things where the model disclosed new information about the mechanisms of the interaction between the pharmacology and the biology?
Yeah, so there were two examples coming to mind. So the first one, as I mentioned or alluded to, was that the model does not have an extensive representation of the epithelial lining or the barrier function. Through what we had seen, we had a wealth of information on the bacterial, such as commensal versus pathogenic, the microbiome of these patients, their genomic information as well. And we found that that played a big role in the level of, let's say, TNF alpha or IL-6, the level of macrophage activity varied quite a bit between different patients and seemed to be correlated strongly with these bacterial populations. So that was an immediate gap we recognized. And the way we accounted for that is by implementing a different hypothesis, what we call the necrosome autoamplification loop. So what we had found in the literature was that the epithelial cells, once they're exposed to, let's say, pathogenic bacteria, other damage associated molecular patterns, they start to go through necrosis, which is a highly inflammatory way for a cell to die, and it starts to just release everything into an environment. And so that seemed to explain the speed and the rapid onset of inflammatory mediators levels that we were seeing in some of these patients. So that was something we discovered along the way. And the other example I was going to bring up was that the other key part is the Tregs, which again I alluded to, was that for some patients, they had high levels of Treg in their biopsy samples, yet they still had a very severe disease. So did this mean that the Tregs are somehow quieted? Were they not serving their purpose of immunotolerance? So this was still an ongoing question we had. And the way that we had addressed it is by differentiating between local Tregs in the lymph nodes. As you know, that the gut system has its own kind of special lymphatic system. There's already a lot of memory Tregs, natural Tregs kind of in waiting. So as soon as inflammation happens, they can be rapidly recruited. On the other side of it was some patients didn't seem to quite have that kind of rapid recruitment of Tregs, or rather it would be the slow recruitment of other induced Tregs from the body. So that's something we had discovered through the data. So we had implemented kind of a differentiation between, okay, was it an induced Treg, a natural Treg, was it local, was it not, so that's something we had also found out along the way.
So a layman's suggestion, I really think it's important that people doing the kind of work you're doing include that kind of information every time you do one of these briefings. That it's one thing to predict the outcome in statistical numbers, but when you can cite that the process of working the model, we came across some other things and questions that we were able to work with the sponsor or biologists, or questions that we now have in a list that we need to explore, because that is incredibly valuable, and that's discovering new stuff, not predicting old stuff.
Yeah. No, that's a great point. Yeah, no, thanks for that.
Other questions? Connor.
Sure. So sorry if I missed this at all, but I was wondering if you could explain some of the details of the mathematical model. Is it primarily ODEs? Is it a combination of different types of equations? Yeah, just those kinds of things.
Sure. Yeah, it is a system of ODEs. And again, they were primarily representing the activity of the cells. So those things, we had data on the number of cells per milliliter, let's say, like densities of these cells, but we're primarily looking at what's the activity and what's the level or concentration of cytokines that they're secreting. So it was I'd say, yeah, well, it's all published, so we could share with you the Rogers paper is primarily what it's based on. But yeah, it's primarily ODE model with various regulations kind of built in.
I guess to follow up on that, so I mean, you have the extracellular environment in which you have populations of cells,
Mhm.
And then you also have models of specific signaling and response pathways within individual cells.
Right. Right.
And so coupling those layers is non-trivial in a pure ODE model. If you have an agent-based model where you have different representations of different cell types, it's easier, well, I guess you do compartmental models where you have some representation of the typical Treg cell of a particular type, the particular cell of a particular type, and then that interacts. But even then,
Right.
coupling those with population models isn't so trivial. And again, I think QSP people know how to have this in their bones, but for our audience, maybe we don't understand exactly how that works.
Yeah, it's a good question. And I have to be honest, like I'm not the expert on what happens intracellularly. I know there's a lot going on. We've done modeling before of the TNF receptor, tyrosine kinase receptors. Like it's very complicated biology that's occurring, and the kinetics as well. So as you probably picked up from my talk, was that it's a multi-scale model, not just in terms of the intracellular versus extracellular, and then back to the organ level kind of dynamics. But also across time. So we're trying to simulate things on the order of days and weeks. So things like intracellular signaling pathways, we're not as much concerned about. Essentially, the way the model is kind of constructed is we establish these Emax models or dose response relationships between, let's say, the extracellular TNF ligand concentration and what that would do to macrophage secretion of another cytokine. So we have this kind of web of these various dose responses that's kind of baked in. We try and limit as much as we can because we don't want to be adding in too many parameters that we can't quite identify. And honestly, many of these parameters wouldn't be identifiable. But we rely a lot on in vitro studies that have studied kind of these cell types, their migration, their secretion patterns based on different stimuli. And it's not as precise as, let's say, down to the second level, number of seconds or minutes, but it's sort of how we balance the two sides. It's more of an abstraction of what's really occurring. Yeah.
Great. Thank you. That was very helpful.
Sure.
Joel, did you have a question?
Yeah, I think it's a great segue because I was thinking exactly about that, about how you validate not only based on your clinical trials, but the information you find usually in literature is done on in vitro models, right? And having this combination of things, my question would be, when you are calibrating your model, which is there one information that wins over the other when there is any discrepancy on the levels you're seeing on in vitro data versus the result you're getting for your clinical comparison?
Right. Yeah. So we begin with kind of the end result in mind, right? Which is we want to know what's happening inside a human in vivo. So as close as we can get, that's kind of how we decide our data choice. So for example, there's plenty of studies that can be done on human PBMCs, how they react to various cytokines, how they proliferate, how they secrete. So that's a readily plentiful source of data. But we want to know is, are there intestinal T lymphocytes, intestinal epithelial cells, those cell line studies, like those would take precedence over the two. And it's actually quite difficult to match the cytokine milieu, the exact environment of the intestinal system. So there are a lot of studies that try to do this on things like organs on a chip, for instance. But to be honest, that is the exception, right? That's not the normal source of data. So oftentimes we do have to rely on human PBMC's data, but if that is the case, we want to look at an average across different papers to make sure we're understanding the correct relationships, rather than focusing on a single study at a time. We all also aware, different studies, they may even have the same experimental setup supposedly, but they can still have very different results. That's something commonly we see. So in those types of cases, luckily for us, when we're working with the sponsor, we can organize quick studies. This model was built over the course of 3 years, let's say. So we did have we had the time to afford it, let's say, if we had some deeper questions. But still, I wouldn't say that the model is fully identifiable. Not close. Yeah.
Right. Yeah. And I guess, right, animal data in comparison to also human data, right? There's all that. But just out of curiosity, how many people are working on just this project?
So I would say in total, it would have been, in terms of modelers, let's say, it'd be like a team of 5 modelers.
Mhm.
And then in terms of like wet bench, wet lab scientists who are supporting this work, it's a far larger number than we can understand. But that's a nice part is, IBD, there is some relation to other autoimmune indications. So we get to borrow data from psoriasis, from rheumatoid arthritis at times as well.
Great, thank you.
Sure.
Other questions? Maybe if there isn't an immediate one, I would like to come back to something you talked about a little bit and John talked about, which is the idea of personalization. And so, as you point out, the number of clinical markers that you can observe in a patient is fairly limited. And so, if you want to then develop a digital twin of the individual, you're going to have to do a lot of inference and hope. Presumably, you'll actually have some kind of cloud of models of the patient, not a single model of the patient. How practical do you think it is to go backwards and to try to begin to specialize your models? You've done some stratification, so clearly the markers have some stratificatory value.
Mhm.
But how far are you from being able to say this patient? If I were a clinician, I make some observation about this patient, and now I want to pull them into your model and then do forward prediction. How far away are you from being able to do that? What are the key obstacles to it?
Yeah, that's a great question. Off the top of my head, I would say I want to base my answer on the closest thing that I feel like I've seen for a digital twin, and that's actually for neurodegenerative diseases. So it seems like that approach has gone quite close to the digital twin approach, where again, the key output are things like imaging studies, SUVR for amyloid buildup, for tau protein buildup, but they're trying to relate that to what is the actual disease progression, what's the rate of amyloid and tau and those and fibril buildup and things like that. So based on what I've seen, what they're able to do is they start off probably by having, I think, a simpler model is the way to do it. So something that really maps the dynamics with those imaging studies, and then from there, very carefully expanding out based on the other observations that they see. So based off of that, with this model, I think the original purpose of the Rogers model, which is what this model is based on, wasn't really so much the digital twin approach. So it's probably several steps away from getting there, because there are quite extensive interactions between an M1 macrophage and other immune cells in the system, but we don't know what those relationships are really like in vivo inside the gut. So until we understand those types of relationships, I would say we're quite far from digital twin. But as you pointed out, yeah, what we're finding is if we're trying to understand comparatively how will your therapy perform compared to another therapy for a population of patients, we can start to answer those types of questions and try and stratify, will your drug work for a set of patients who are not responsive to another drug, let's say. Yeah.
There you go. So I mean, you talked about the difficulty of going from the molecular markers to the clinical outcomes, and you pointed out that you can have divergence. For example, Tregs, the same level of Tregs could seem to do different things because you don't see the origin of the Tregs or the location.
Right.
And so the forward problem is already hard, and the backward problem presumably is harder.
Yeah.
Something that you could probably do from your virtual patient populations, which would be interesting, and I think I'm sure you're thinking about, is from the digital twin point of view, which is you have, in this case, a lot of different possible clinical intervention strategies. And so one thing you would like to be able to do with an individual patient is to determine, is the intervention strategy working or not, as quickly as possible. And I would think that even with the virtual patients that you have, you could ask the question, if I give the correct treatment or the wrong treatment, given the uncertainties in the system, how long do I have to observe the clinical markers before I can say with some certainty the treatment is working or not?
Right.
I would think that would be a very useful outcome, even if you can't back out the parameters for personalization of therapy. And I wondered how that plays out within the virtual populations that you're looking at, and whether you've looked into that particular question, whether you can use this to basically reject therapies that are not working faster so that you can try different strategies.
Sure. Yeah, no, we've considered it. We haven't quite executed on that idea yet, but there is actually an interesting paper that came out, I believe it was last year, 2021, where what they did was they sampled the parameter space very extensively, a lot more than what we have done. And they start to track a real life patient and match up as close as possible to which of those parameterizations this real life patient most closely matches with time as it goes forward. So once it reaches week 12, they select the appropriate virtual patient that most closely resembles, and they start to try and answer those types of questions, whether this patient is on the right track and will continue to respond out to week 52, or whether this patient is likely to build resistance to the therapy and rebound to severe disease again, by 1 year's time. So that's a quite interesting idea, and that type of work is being done at the moment.
Joel, another question.
Yeah, the what James just mentioned is I know that you mentioned a little bit of how heterogeneity is determined on your model, but how many parameters is that you're like having a range of possible values? How actually you determine all the heterogeneity of your virtual patients?
Yeah, so that's a great question. So we have a limited set of model parameters that we vary. We sample from, let's say, a log normal distribution, and we don't quite know. We know based off of, we start off by tuning a reference patient or a nominal patient, let's say, that hits all the mean measurements. But we don't quite match the distributions yet for these outcomes. So we start to sample around key parameters, following a sensitivity analysis. We understand what are the most sensitive parameters for the given output. We start small with a minimal set and start to expand as we go. And the range that we're sampling from, there's no way we can infer what that range is, but we start off with a sensible range and we start to see how the virtual population is filled out. There's actually quite a nice approach that's been developed by Richard Allen over at Pfizer, which you're familiar with, which is kind of this, it's kind of Monte Carlo based, like what I'm talking about. But ultimately, we're trying to find individual patients that match the distributions for several measures all at once, using the same underlying QSP model and the same model parameters that are being varied. The key part, though, there is, making sure that not just the known distributions are matched. We also want to look into the unknown to make sure there is no what we consider an unphysiological or unhuman patient, right? Where there'd be some marker that's, something that, if a scientist sees it, they'll start laughing. That doesn't work. So so there's some mental checks, some visual predictive checks that go along the way. But that's actually quite a extensive practice or project, to get that work done. And there's actually some good research being done in that area as well.
I was going to say, I mean, a lot of people in our small community do spatial modeling. So, for example, Joel and I do models of epithelia where you have each cell represented explicitly. Of course, they have a lot of parameters. They're hard to identify. But then you can begin maybe to address some of the questions about barrier function and spatial. I mean, lesions in the gut are localized, for example. And so you'll have different immune profiles in different positions within the gut, as you pointed out. So those are the kinds of things that when you don't need that kind of detail, of course, you're much better off not using it, but sometimes you need it. And so it's quite interesting to see you sort of identify as a missing piece the specific kind of thing that many of the people in our community do, which is spatial modeling of, especially of epithelia. And so it would be fascinating to see how one could take the kind of structural models that we're used to and put in the very, very detailed molecular understanding that you have. And I think those are potentially interesting directions to move for some of this community.
Yeah, absolutely. Yeah, we know in the data that there are differences between different locations of the gut, how the IBD progresses. So it may come down to differences in epithelia, but that'd be quite interesting. Yeah.
Well, even the microbiota change from millimeter to millimeter within the gut, right? You have these loci.
Absolutely.
So uh, uh, I always find the gut microbiota terrifyingly complicated. The human body is already complex enough, but the gut microbiota really get elaborate. So if you're able to do predictions including that, that's really, to me, very impressive. Are there any other questions for our speaker, or final questions before we break? Well, I want to thank you again for coming in. I think we've had a good set of questions and discussions. I really do appreciate this. I put in the chat some of the links on the slides so that people can look up the papers that you're referencing.
Right.
And I hope we'll hear more from you and other members of your team as things progress about how to, especially into the area of personalization, and how to apply these methods in the context of digital twins and model personalization. So thank you, everyone, for coming, and thank you for speaking today. And we'll see you again in 2 weeks then. Goodbye.